That's tuberculosis, okay, where the TB bacillus gets all the benefit because it grows and multiplies and spreads its range. And of course, the host gets very, very sick, okay? So the human body has a lot of different environments because it has a lot of different cavities. We have the surface of our skin. We have our mucous membranes. We have the peritoneal and the thoracic cavity. We have the lungs. We have the entire urinary and digestive system. All of these that are open to the outside world are going to be populated with some kind of microbe, okay? We're in an equilibrium with these microbes. If we're healthy, we live with those microbes and both remain healthy because it's a symbiotic relationship, but sometimes we generate a colony of pathogens, one or more, that ends up producing disease, okay? And that's often because our beneficial bacteria get crippled somehow, okay? Or we get exposed in a way that we shouldn't be. So the Human Microbiome Project was an effort to try and figure out what is living on us, in us, and between us.
All right,
And
okay.
hold on.
Because if she don't have her card on it, then I ain't even gonna take my break yet.
I want to say it might the most, I think it's 27.
What y'all looking for?
So what's living on us, in us, and between us, right? And by describing it as a microbiome, what we're saying is that there's a variety of different bacterial species within that environment, right? They're all interacting, they're all vying for space, okay? The microbiome is going to be different from person to person. And the reason is that every person is a little bit different genetically, okay? And in how they interact with the environment around them. The way that we know what's growing on us, around us, and in us is the use of DNA analysis. We have a way to isolate genetic material from any sample and amplify it, and then look at the information in that DNA and see if it looks like the information from certain microbes, okay? Human cells have about 22,000 protein coding genes. Our microbiota, which are our guests that are living with us, about 8 million, okay? So that's a lot of extra information, right? Microbes are found in locations that we used to think were sterile. All healthy people have dangerous pathogens, but they're present in tiny numbers, and that's why they're not getting sick, okay? So the makeup of our biota on us, in us, and around us is going to have an impact on our health, right? So we have a lot of different environments inside the human body, right? We've just named a few of them, right? The urinary tract, the digestive system, the respiratory system, the surface of our skin, and so on, okay? Most areas of the body that are in contact with the outdoor environment, external environment, are going to have microbes in them. Internal organs and tissues and their fluids should be microbe-free. Note I say should be. If they've been damaged from trauma, they're going to be infected, okay? The majority of microbes that come in contact with the human body get destroyed by our immune system before they get a chance to grow. The microbes that remain have a relationship with the host where they're able to survive and not hurt the host, okay? So you can see on the right in table 11.1, some different environments that we provide, okay? So how is it that we get colonized? Well, we get colonized really before birth, okay? Because again, the womb is not isolated from the outside environment, right? It's connected via the birth canal and the vagina to the external environment. So the microbes can get up in there, okay? The normal biota are unlikely to be displaced by incoming microbes because there's nowhere for them to hook up, because the The good bacteria, the good microbes have already done that work. The chemical and the physiological environment that's created by resident biota is hostile to other microbes. They have a way of, for instance, peppering their environment with compounds that retard the growth of other types of microbes, right? I'm here first and I stake out my territory, okay? Normal biota is beneficial or at worst commensal to the host in good health with a functioning immune system. Factors that weaken that immune system are going to increase our susceptibility to infection. So what would these conditions be? Well, these are a few of them, right? Old age or extreme youth. Now, why the two extremes? Well, because in old age, in every organ system, cell death exceeds the rate of cell reproduction. And so tissues, any type of tissue, becomes less functional, which means there's fewer white blood cells, for instance, to defend against attack. There are going to be fewer immunoglobulins to defend against attack. Okay, the liver itself is going to be less capable of producing complement, which helps the immune system to destroy pathogens. Okay, just plain old tissue breakdown, right? The reason extreme youth is dangerous is because the immune system hasn't fully developed yet. Okay, remember that the thymus is the home of the development of cytotoxic T cells, which are white blood cells that attack infected tissue and destroy them. Well, in a newborn, the selection of those cells and the deployment of those cells hasn't happened yet. Okay, so that part of our immune system hasn't graduated from thymus college. Okay, genetic defects in immunity and acquired defects in immunity can weaken our defenses. Okay, genetic defects would be things like inheriting a gene that makes us unable to produce certain white blood cells or any white blood cells. Okay, acquired immunity would be defects in immunity, be things like AIDS or chemotherapy, right? The result of attacking the white blood cells is to reduce the ability to fight infection. So you get what are called opportunistic infections. And let's write that down. Opportunistic infections. Okay. Opportunistic infections. Surgery and organ transplants can be damaging. Again, why? Because frequently when we do like an organ transplant, we have to use immune suppressants, and that leaves you open to opportunistic infections. Surgery opens the inside of you up to the outside, and so it can get contaminated. All right. Then there's underlying disease such as cancer or liver malfunction or things like diabetes, which are going to weaken immune function. All these weaken immune function, immunity. Okay. Immunosuppressive drugs, chemotherapy, right? Stress will do this as well. One of the main hormones that does this is called cortisol. Okay. Cortisol elevates blood sugar, but it also is going to reduce inflammation. Okay. And inflammation is critical to fighting disease. And then pregnancy, because it stresses the mother's immune system. Okay. So until 2013, we thought the uterus and anything inside it was sterile. Okay. But newborn stool shows that they're colonized before they're born. Okay. And so this means that their intestines get colonized while they're still in the uterus. Not a surprise, really, because the birth canal connects to the outside. Comprehensive exposure occurs during the birth process. Within about a day, a vaginally delivered baby has been colonized by lactobacillus, Prevotella, and Escherichia, right? The lactobacillus provides the baby with enzymes to break down milk, and other colonizers protect the baby from skin disorders like staph epidermis would be an example. S. epidermis. Okay. Human milk has about 600 species of bacteria. You might say, well, how'd that happen? Well, remember that the nipple has an opening, right? Which means that that's a port of entry, which means bacteria can get in there, okay, into the mammary glands, right? This project showed that there were significant variation in our normal microbiota. The biota on a right hand can be different from the biota in the same person on the left hand. It's more important than the exact microbial profile is the protein and enzyme profile of our microbiome. Okay, that's what this is. The microbiome is basically the living microbes with us. Okay, all of them. There's about 100 types of fungi in the intestine, 1 billion viruses in the feces. Okay, so all of that's there all the time. All right. So we're going to talk about how pathogens are able to create disease, right? What is it about a pathogen that makes it capable of generating a condition that makes us sick? Okay. All right. So a pathogen is just something that produces disease, right? Pathogens can either be living or non-living. Non-living would be things like viruses and toxins and prions. Okay. And then living pathogens would be bacteria, mold, okay, or you could say fungi if you like. Okay. And protozoa, and then even parasites. Okay, living pathogens. Okay. So it's the way it makes its living is it produces disease, and during that process, it's able to gain a place to live. It's able to gain nutrients and building blocks, and it's able to grow, right? Disease literally means not at ease. Not is from dis and ease is ease, okay? An infectious disease is a disruption of a tissue or organ caused by the microbes and what it makes. The type and the severity of an infection depends on the pathogenicity of the organism and the health of the host, right? Pathogenicity is just an organism's potential to cause disease, okay? And pathogenicity is enhanced by what are called virulence factors. You could think of virulence factors as weapons. To make you sick. Will. Okay. A true pathogen can cause disease in a healthy human. And even though the immune system is present, it's able to slide past all that stuff and generate a disease. Okay. A lot of these vary in severity from mild to fatal. Examples: flu virus, plague bacillus, malarial protozoa. Okay. Here, so this is a eukaryote protozoa. Bacillus is a prokaryote, and the virus, of course, is non-living, right? So the way that we determine whether or not disease is produced, all right, we have to have the set of conditions down here that meet those qualifications, right? We have to have a high virulence for the microbe. We have to have a high percentage of the infectious dose, which is the number of microbes that need to get in you and all over you in order to make you sick. We have to have a genetic profile that resists microbe X not present, okay? And then we have to have a level of health that can be anywhere from healthy to suppressed, okay? And that's going to generate disease, okay? Those are the disease conditions. Any deviation from that, we don't get disease, right? All these other situations either lack the virulence or lack the number of bacteria, or they lack the genetic susceptibility to the disease, right? Or healthy individuals basically picked off those pathogens before they make you sick, okay? Opportunistic pathogens are what make disease when you're healthy or when you're not, when you're immune suppressed, okay? So act on immunosuppressed targets, okay? They make the disease when the host defenses aren't working the way that they're supposed to. They're not pathogenic in normal, healthy individuals, and they don't have really well-recognized virulence factors, right? They don't really have weapons to produce sickness that would slice through a healthy defense, right? Examples include Pseudomonas and Candida, okay? If you work with patients, for instance, that have, say, liver damage, or patients that are on chemo, okay? One of the things that could sometimes develop is thrush. And when you look at somebody with thrush, their tongue is covered in white fuzz. And that's the growth of Candida, which is a fungus, okay, on the mucous membrane that normally wouldn't occur because the immune system would keep it in check, okay? So that's just one example. Virulence is just the ability to produce disease. Something that's highly virulent produces disease very easily. Something that's not virulent does not, okay? So it's the degree of pathogenicity. So how do we determine their virulence? Well, its ability to establish itself in the host, its ability to damage the host, right? A virulence factor is a character of the microbe that contributes to its ability to establish itself in the host and make damage, okay? Remember that we call these weapons of illness, weapons to make illness. Okay. Weapons to produce illness. The port of entry is just the way that the microbe gets in you, okay? The route of a microbe into the body has to be appropriate for it to produce disease, okay? So for instance, something that causes a respiratory infection might not produce sickness if it comes in through the urinary system, okay? Have to have the right door to go through in order to produce the illness, because that leads to the area of the body that can be colonized, right? Exogenous microbes come from a source outside the body, which is the environment, or something in the environment, like another person or an animal or an object, okay? Whereas endogenous are microbes that are already on you, in you, and around you, and were kept at bay by your normal biota, right? But what happens then is they can emerge if your normal biota get messed up, or if your immune system is not working the way that it's supposed to, okay? Portals of entry are generally through organ systems open to the outside world, meaning they're lined with mucous membranes. So Examples of those organ systems: respiratory, digestive, urinary, reproductive. Okay. All of those have natural portals of entry. Other portals of entry: the nipples, the tear ducts, okay, the external acoustic hiatus. This is the ear canal that leads back to the drum. Okay. The eustachian tube. Okay. And then any cut, puncture, or abrasion on any of these structures or on the intact skin or mucous membranes, right? Those are all ways we can get in. Once we're in, then we can cause trouble, okay? An infectious agent can sometimes get inside the body by more than one doorway, right? TB can enter through the respiratory and the GI, right? Strep and staph can come in through the skin, the urogenital, or the respiratory, okay? The infectious dose is just the minimum number of microbes that causes the infection to take off, okay? So you can see that if you're below the infectious dose, you're likely not going to get sick, right? But the lower the infectious dose, okay, the higher the pathogenicity. Okay. So if my infectious dose is, say, a single cell, like for rickettsia, we're talking extremely pathogenic microbes. Okay. You can see that infectious dose varies, right? The first step in producing infection is to latch on to a surface so that you can begin to grow, right? They gain a more stable foothold on the host, depending on the specific molecules that the host has. The pathogen can either hook up or not, okay? Pathogens can be limited to only those cells and organisms to which it can bind. If it can't bind, it can't take off and grow, right? Because it's going to be flushed out of the system. So how do we do this, right? Well, we have extensions on the microbes, such as fimbriae and pili and other proteins that can interact with things like receptors on the surface of our tissues, right? Firm attachment is a prereq for making disease, since the body has so many mechanisms to flush out what comes in, right? So those are some of the mechanisms for hooking up, right? You've got all kinds of ways, right? Receptors, capsules, surface proteins, and even in worms, things like suckers and hooks, okay? Nasty stuff. Phagocytes engulf and destroy host pathogens using lysosomal enzymes or antimicrobials, right? Antiphagocytic factors are basically weapons, virulence factors, that help the pathogen to avoid the phagocyte. Like, for instance, an example would be leukocytin, right? It kills the phagocyte and makes it difficult for the phagocyte to do its work, right? It either can't grip and engulf the pathogen and kill it, or the phagocyte commits suicide before it gets a chance to do its work, okay? So those are a couple of examples, right? The slime capsule can make it tough for the phagocyte to grip the microbe and eat it. And some bacteria can actually grow inside a white blood cell, okay? Pretty nasty. Virulence factors are just adaptations that the microbe uses to grow inside the host. Three ways that microbes damage the host is through enzyme activity that tears up stuff that we need, and through the action of toxins. Okay, there's two kinds of toxins. There's endotoxins, all right, and there's exotoxins. Endotoxins are attached to the surface of the pathogen. Exotoxins are secreted. Okay, and these are bound, right, bound to pathogen. Okay, and there are some pathogens that have both. So this indirectly can induce the host defense to respond in an inappropriate fashion. Okay, you might say, well, give me an example. Well, we're going to learn a little bit later on that there's a little microbe called a spirochete. Okay. Spirochete that I'm thinking of causes something known as Lyme disease. Okay. What the spirochete does is he just kind of wriggles his way through tissues and between tissues. And as he does, he leaves behind antigens like a trail that trick the immune system into thinking that those cells are infected. The result is that the immune system attacks and destroys those cells, and that serves as a decoy to let the spirochete escape. Okay, so that's one example. Exoenzymes are secreted by microbes and they break down or damage tissue. They can dissolve the host defense barriers and promote the spread of disease. Examples of these include mucinase that tears up the protective coating of mucous membrane, hyaluronidase that tears up ground substance that's part of connective tissue in the body, and coagulase that causes the clotting of blood or plasma, and kinase that dissolves fibrin, which is what clots are made out of. A toxin is a specific chemical product of the microbe and some animals that poisons the target, poisons the host, right? Neurotoxins hit the nervous system, enterotoxins the GI, nephrotoxins the kidneys, hemolysins attack RBCs, red blood cells. An endotoxin is attached to the pathogen. An example is lipopolysaccharide, which is part of the outer membrane of gram-negative cell walls. So everywhere that gram-negative goes, that endotoxin is going to produce disease, damage, okay? It can cause fever, inflammation, hemorrhage, diarrhea, right? Blood infections by Salmonella, Shigella, Escherichia, and E. coli are also dangerous, right? So you can see some examples of different exotoxins and endotoxins, right? And how they do their work. How do we spread the pathologic effect? Well, we settle in a target organ and we colonize it. We damage the site of attachment. The tissues get weak because the pathogen reproduces on their surface or inside them. The pathogens can block tubes, such as blood vessels or lymphatics or fallopian tubes, and that's going to cause disease, right? Block flow. Necrosis is accumulated damage to tissue from pathogens that leads to tissue death, right? Viruses can destroy cells by multiplying inside them and rupturing them from the inside out, right? So you can see some examples here of different types of infection. This is 11.4 in your book, but below you can see the process, right? You got to have a port of entry. You got to latch on. You've got to resist our defenses, and then you make disease. And then at some point, or sometimes not, you exit the host. Okay, most of the time you exit the host. You could stay in the host and exit the host, right? You can have pathogen reproduce. Some pathogen stays behind. Some pathogen exits the host. Okay, so again, it all depends on the pathogen that we're talking about. A sign is an objective measure of disease that we note, and it always has a number associated with it. We can put a number on any sign, okay? As a numerical value, sorry, numerical value. Okay. A symptom is evidence of disease described by the patient, thing like pain, for instance, okay, or discoloration, right? And a syndrome is just a collection of signs and symptoms indicative of a particular type of disease, okay? Inflammation is a normal response to infection. Symptoms include fever, pain, soreness, swelling, okay? Signs, edema, which is the accumulation of fluid in the tissue, okay? Granulomas and abscesses, which are collections of inflammatory cells and microbes in the tissue. Lymphadenitis, which is swollen lymph nodes, okay? And again, note the difference between the two, right? Symptom is subjective, okay? And a sign is objective, measurable, okay? So here, fever, pain, soreness, swelling, we can put some numbers on this, like we could probably put a number on fever. We make up scales for pain, okay? But generally we just ask, you know, or observe whether they have those conditions. But a sign, we can put a value on, right? Edema, swelling, right? We could put a number on how swollen. Okay. Granulomas and abscesses, the size and distribution. Okay. Lymphadenitis, number of affected nodes. Okay. So we can put numbers on all these things. Blood infection. Examples include leukocytosis, where you have elevated white count, leukopenia, which is a decrease in white count, and septicemia, in which microorganisms are multiplying in the blood and are present in large numbers, and bacteremia and viremia, where the bacteria or viruses are in the blood but are not reproducing. Okay, so emia means in the blood. In the blood. Okay. And then what comes before it is the descriptor, right? Bacteremia, bacteria in the blood, viremia, viruses in the blood, septicemia, septic components in the blood. Okay. So that's how we use the terminology, right? Some infections go unnoticed, right? The microbe is present in the host tissue, no symptoms are produced, no medical attention is sought, and these infections are called asymptomatic, subclinical, or sometimes referred to as inapparent, right? We don't see them. The danger, though, is that even these individuals can spread disease, okay? So let's take a break. Let's take 15 minutes. I'll see you guys at 2 o'clock, and then we will pick up with the next half of this chapter. Okay. Moving on. If we get in, we eventually get out, right? Although in some cases, the pathogen will stay with you your entire life. A good example of that would be viruses, okay? The portal of exit is an area where the pathogen leaves the host. It can be through secretion, excretion, discharge, or sloughed tissue. A high number of microbes in these materials increases the likelihood that the pathogen is going to reach other targets, right? The portal of exit is usually the same as the portal of entry, but sometimes it's different. And you can see some examples of exit portals here, okay? Sometimes a pathogen can hang with you for most of your natural life, right? Latency is a dormant state of microbes in certain chronic infectious disease. Viral latency includes herpes simplex, zoster, Hep B, AIDS, and Epstein-Barr. Hold on a second. Okay, I'm back. An example would be viral latency. What happens in a lot of viral infections is that the genetic material from the virus can integrate into our own genetic material and remain there for the rest of your life. Okay, an example would be herpes zoster, right? This is the agent of chickenpox and shingles. If you get chickenpox, the virus places its genetic material in the nucleus of nerve cells where drugs don't get very well and remains there for the rest of your natural life and will reemerge when the immune system is somewhat suppressed. Okay. Other examples, Hep B, AIDS, Epstein-Barr can all go latent. All right. Bacterial or protozoal latency includes syphilis, typhoid, TB, malaria. Okay. And this could be from spore formation. This could be from simply living within the host tissues without actively reproducing. Okay. Or it could just be low levels of pathogen that occasionally reproduce to high enough levels to produce disease. Okay. Sequelae are long-term damage that are caused by these types of infection, right? So even though you might clear the pathogen, the damage left behind never goes away. Examples include deafness from meningitis, rheumatic heart disease from strep throat, Lyme disease causing arthritis, and paralysis coming from polio, right? That stuff doesn't get fixed, unfortunately, okay? So again, another reason to try and control where the pathogen can go. The incubation period is simply the time from initial contact with the infectious agent to when the symptoms happen. At this point, the pathogen's multiplying at the portal of entry, but has not caused enough damage to elicit symptoms. This varies according to host resistance, the degree of virulence, and the distance between the target and the portal of entry, and ranges from several hours to several years. The majority of infections range from 2 to 30 days. In the prodromal stage, there's a couple of day period where the earliest notable symptoms appear. You have a vague feeling of discomfort, head and muscle ache, fatigue, upset stomach, and malaise, right? The period of invasion is where the infectious agent multiplies at increased levels. We see toxicity. It gets well established in the host. We see fever and other signs and symptoms. This period is variable depending on the pathogen, depending on the disease, okay, and the condition of the patient. The convalescent period is when the patient begins to respond to the infection and the symptoms begin to decline because the immune system has now kicked in. The patient's strength and health return because of the healing nature of the immune response, and the patient often patients will quit taking antibiotics during this time, but they can still spread the disease. So this is one of the ways we get drug-resistant strains of bacteria, all right? Very dangerous. The incubation period is a time during which pathogens such as measles can be released, while the invasive period is when Shigella can be released and Hep B can be released at all times, right? So you can see an example here of these different stages of infection. And as we stated, the windows for each of these can vary, all right? But you don't notice anything until when? Until you get here, right? The period of invasion and the height of the infection. In all these other stages, right, incubation, prodromal, convalescent, symptoms are declining, and that's often when you can spread disease to coworkers, friends, and family, right? Because you think you're either not sick or you're over it, okay? Now, pathogens like to hang in areas that are called reservoirs, just like a water reservoir is where we get spare water when it's unavailable from other sources, okay? This is the primary habitat in the environment where the pathogen comes from. It can be a human or an animal. It can be soil, water, plants, all kinds of areas where the pathogen can hang out, okay? A transmitter is just someone or some object where you can acquire the infection, okay? Syphilis are the same for the reservoir and the transmitter. It's the human, okay? Hep A, the reservoir is the human, the transmitter is food, okay? A lot of infections that hit humans have reservoirs in other humans, persons or animals with symptomatic infection or obvious sources, but you can also get it from individuals that are prodromal or individuals that are asymptomatic, okay? Asymptomatic carriers, examples of spread can be gonorrhea, genital herpes without lesions, or human papillomavirus. An incubating carrier could be somebody with mono, convalescent carrier, something like Hep A, or a chronic carrier could be TB or typhoid, right? The carrier is just someone who has the pathogen on them or in them and spreads it to others, and they don't notice it because they haven't experienced symptoms, right? But they have the microbe. Animals can be reservoirs and sources, right? Animal reservoirs are agents like, hold on. All right, we're back. Okay. So animals can be reservoirs and sources. Okay. And again, whether you're a reservoir or a source is going to depend on the ability of the pathogen to attach, to colonize, right, and to grow. Animal reservoirs, arthropods like fleas, mosquitoes, ticks. A biological vector is going to participate in the life cycle of the pathogen, while a mechanical vector is simply going to carry the pathogen from one place to another on its body parts, right? For instance, a fly is a mechanical vector for a lot of disease, right? It can land on something that's contaminated. It can pick that up on its feet, and then it can land on something that's uncontaminated, right? Whereas a mosquito, for instance, can carry um, diseases such as malaria, all right? And the parasite spends part of its life cycle in the mosquito, and the mosquito could transmit it then to a target like a human, okay? Larger animals can spread infection, rabies from mammals, pisticosis from birds, and salmonella from lizards, okay? A zoonosis is just an infection that is indigenous to animals but can be naturally transmitted to humans. Humans are essentially the dead end host that don't contribute to the natural persistence of the microbe. Some zoonotic infections have multi-host involvement. Some have complex life cycles. Zoonotic spread is promoted by close association between humans and animals. So thinking what? People who work in kennels, people who work on farms, okay? People who work, say, in the meat industry, all right? Those are all areas where the close association between the human and the animal could result in spread, okay? Non-living reservoirs, things like soil and water, okay, or foods, okay? Most microbes that thrive in soil and water are saprobic and don't harm humans or have little benefit, but some are pathogens and some are opportunistic infections, okay? Because humans are in contact with these sources, you can pick up the pathogen from a natural habitat. And so this helps us in the analysis of the spread of disease, right? Where have you been? What have you interacted with? And that can give us a clue as to what you picked up, okay? In addition, of course, to looking at the signs and the symptoms and, of course, examining infected material under the scope, right? A communicable disease is one where the infected host can transmit the agent to another host and it can establish an infection there. And infectious and communicable really are interchangeable, right? While contagious is a disease that's very communicable, okay? Flu and measles are highly contagious, whereas leprosy is only weakly contagious, okay? And a non-communicable disease is an infectious disease that doesn't arise from transmission of the infectious agent from host to host, right? An example would be somebody who's immunosuppressed and has candidiasis or thrush, right? It's growing on them because their immune system's not up to snuff, but they can't spread that infection to you because you have a healthy and functioning immune system, okay? Or it could be someone in accidental contact with a microbe that comes from a non-living reservoir, right? So they're not a source of disease to other people. How can we transmit? Well, there's a variety of ways, okay? Direct or indirect contact with an animate or an inanimate object. Animate object, this means it's alive, okay? So you pet your dog or you stroke your cat or you touch a snake or a lizard, all right? Or another human, right? And that could have the pathogen on it or in it. In horizontal transfer, the disease gets spread through the population from one infected individual to the next, while in vertical, the disease is transmitted from parent to offspring. Two major modes: direct contact and indirect transmission, where you have a vehicle. Okay. So you can see some examples here, right? Direct contact. Okay. You're right there in the person's local environment, and you either touch or you aerosol or you're spread by some other method. Okay. Those are examples of direct contact. Indirect transmission. You're infected, you touch something, somebody else touches that same thing, they pick it up. Okay. That's just one example. And then vector transmission, we've got mechanical and biological. Okay. And we got the housefly there and the mosquito. And then we have infections that are associated with clinical environments. And these are called nosocomial infections. Okay. Nosocomial means associated with a clinical environment. Right. So let's indicate that. From a clinical environment. Okay. That includes infections picked up in a hospital stay or in a free clinic, for instance. Okay. As many as 20% of all admitted patients with an average age of 5%, okay, 5 years, are going to pick these up. 2 to 4 million cases annually, 90,000 deaths, 8 million in additional days of hospitalization, and an increased cost between 5 and 10 billion. So these are nothing to brush off. Okay. Factors: immunocompromised patient. This is a place where a lot of sick people go. So in comes the pathogen with the sick person. Reduced defense allows normal biota to get into the body. The infection can be acquired directly or indirectly from a fomite, from medical equipment, from other patients, from medical personnel, from visitors, from air and water. The healthcare process can cause infections, such as instruments like respirators and thermometers can introduce pathogen. Indwelling devices like catheters or tracheostomy tubes can produce another port of entry. An increased proportion of the hospital population gets antimicrobial therapy, and the result is that you can generate drug-resistant microbes. Okay, so an example of that is something you've probably heard before, MRSA. Okay, MRSA stands for methicillin resistant Staph aureus. Okay, which means it's a type of staph that methicillin doesn't kill. Okay, methicillin is a cousin of penicillin. All right, and this arose because of the improper use of methicillin. Right, use the methicillin until you feel better, put it back on the shelf, go out and spread the resistant leftover bugs to other people. They go to the doctor, they get the methicillin, they repeat the process, and you keep selecting and selecting until you get a pathogen, a strain of that pathogen that cannot be wiped out by that drug. Okay. And then, of course, we know they can share that resistance with other targets. Okay. So what are some examples of healthcare associated infections? UTIs, surgical infections, gram-negative intestinal biota cultured in more than half of the patients that have nosocomials, right? Gram-positive bacteria and yeast make up the rest. Okay. True pathogens such as Mycobacterium tuberculosis, Salmonella, Hep B, and flu can also be transmitted. Okay. How do we control? Well, Medicaid and Medicare don't cover these. Okay. But you do usually have an infectious control officer, and their idea is to monitor this information and put into place procedures that track where the outbreaks come from, identify where it happened, and reduce the occurrence of it in the future. And you do that usually by training and usually by essentially re-engineering how the floor works, okay, and how things are done. Nurses and caregivers are regularly exposed to needle sticks, infectious secretion, blood, and physical contact with patients. So they need to be aware of infection control. So what we want to do at this point is we want to learn a little bit about how to control the spread of disease, okay? And this is going to walk you through that.
Touching hearts and strengthening lives since 1904. Bethesda Lutheran Homes and Services Incorporated provides supports, resources, and worldwide outreach to people with developmental disabilities.
Workers in many different occupations are at risk of exposure to infections. Examples of those at risk are people who work in housekeeping, maintenance, laundry, direct support, or anyone in the healthcare field. To minimize the risk of infection, it is important that everyone follows standard precautions. To understand what standard precautions really means, let's take a look at how the Centers for Disease Control and Prevention define standard precautions. Standard precautions represents a system of barrier precautions to be used by all personnel for contact with blood, body fluids, secretions, excretions, non-intact skin, and mucous membranes, regardless of the person's diagnosis. This system includes the concepts of body substance isolation, which reduces the risk of transmitting pathogens from moist body substances, and universal precautions, which reduces the risk of transmitting bloodborne pathogens. There is a rising concern about exposure to blood due to bloodborne pathogens. You're probably asking yourself, what is a pathogen? Pathogens are disease-causing microorganisms that are found everywhere in the environment, on humans and animals. Pathogens found in the blood are called bloodborne pathogens. Some examples of bloodborne diseases are hepatitis B, hepatitis C, HIV, and malaria. Standard precautions is an approach to infection control that treats all human blood and certain body fluids as if they are infectious. Since these pathogens are too small to be seen by the naked eye, and you can't tell by looking at an individual whether they have a disease, you should protect yourself with simple and effective barriers. This is the rationale of standard precautions. Transmission-based precautions, in addition to standard precautions, are used when someone is suspected of being infected with a highly contagious pathogen. Transmission-based precautions are required where airborne, droplet, and contact transmission of infectious organisms may occur. Transmission-based precautions would be used if a person has chickenpox, measles, or an infection, to name a few. It is not always possible to know whether or not a person is infected with an infectious disease. Universal precautions is made up of a system of safeguards designed to help protect you and the people you support. Safeguards are things like personal protective equipment, safe work practices, good hand washing, good housekeeping, and vaccinations. All of these systems should be used together to form a protective barrier. We'll discuss these safeguards in more detail later. There are some people who may have an infectious disease and not show any symptoms or even be aware of their infection. For example, how many of you know the story of Mary Malone, also known as Typhoid Mary? Mary was the first person found to be a healthy carrier of typhoid fever in the United States. Typhoid fever is a life-threatening illness caused by the bacterium Salmonella typhi. You can get typhoid fever if you eat food or drink beverages that have been handled by a person who is shedding Salmonella typhi. Mary earned her living as a cook for private families. When several outbreaks of typhoid fever developed in homes where she worked, the public health officials questioned her. Under questioning, she stated she rarely washed her hands when cooking and felt it wasn't needed. She also insisted she never had typhoid fever and had always been healthy. However, the health officials linked the outbreaks back to her. After her death in 1938, The autopsy revealed that her gallbladder was still actively shedding typhoid bacteria. This example helps us to understand the importance of following standard precaution procedures at all times. Somehow, pathogens need to get inside your body to cause an infection. Let's take a few minutes to discuss how these pathogens can get inside your body. It can happen from a sharp object contaminated with the infectious material, such as a needle, broken glass, wires, or any other object that can break or cut your skin. From openings in your skin, this would include cuts, nicks, abrasions, skin infections, acne, and surgical incisions. Through your mucous membranes. Mucous membranes are the moist surfaces of your mouth, eyes, and nose. By touching contaminated environmental surfaces and transferring the contaminant to mucous membranes or open skin, surfaces can be heavily contaminated without visible signs of contamination. Diseases that are transmitted from other people or animals are called communicable diseases. Some examples of communicable diseases are measles, strep throat, and colds. Standard precautions should be used in the care of all people and used regardless of whether or not the person is known to have a communicable disease. In order for a disease to spread to others, certain conditions are needed. This is sometimes called the chain of infection. Let's take a few minutes to discuss these conditions. First of all, there must be an agent like a virus, fungus, or bacteria that can cause the illness. The agent also must have a place to live. This is called a host. Humans, animals, and insects are the most common hosts, although agents can also live in the environment. The agent must also have the right environment to survive. Often moist, dark places help them to live and grow. The agent must be able to enter the host. For example, a person who has a break in their skin or someone who breathes in the agent. An agent must have a way to move from its host to infect others. This is called transmission. Some examples are through blood, body fluids, a sneeze, or a cough. There also must be a person who can catch the disease. If someone can't fight off the germ, they can become infected. This is called being susceptible. This completes a chain of infection. The cycle continues as the agent is passed on to another host. The easiest way to remember how diseases can spread is through the David method. Communicable diseases are mainly spread in the following ways. Direct. This is through person-to-person contact. Airborne. This simply means through the air. These infectious droplets remain suspended in the air because they are carried by microscopic droplet nuclei or dust particles. They can land on or be inhaled by the host. Vector. This occurs when an organism, such as a tick, carries disease-causing microorganisms from one host to another. Indirect. This occurs when microorganisms are transmitted by contaminated items such as food, water, medication, and equipment. In other words, person, object, person. Droplets. When someone coughs, sneezes, or talks, tiny droplets may be generated. These infectious droplets propel a short distance through the air and land in the host's eyes, nose, or mouth. Droplets do not remain suspended in the air. The spread of infectious diseases can be stopped by killing the agent, changing the environment so the agent can't survive, wearing protective gear, removing the agent's means of spreading, and making sure people are immune to the agent through immunization. This is commonly called breaking the chain of infection. The most important and effective thing you can do to help prevent the spread of infection is to wash your hands. Hand washing is often taken for granted because it can be routine, but good hand washing stops the spread of contaminants from your hands to other parts of your body or other surfaces you may come in contact with. Let's take a few minutes to go over some general guidelines to follow when washing your hands. If you remove your gloves, wash your hands thoroughly. If skin comes into direct contact with blood or other infectious material, wash with soap and water as soon as possible. If you are using antiseptic towelettes or hand sanitizers, you should still wash your hands as soon as possible after touching something that is visibly contaminated. Remember, good hand washing doesn't take a lot of time. Follow these 6 steps for good hand washing. Remove any jewelry you are wearing, if possible, before washing your hands. Bacteria can thrive under rings and watches. When washing your hands, use warm, running water. Wet your hands and apply soap. Use plenty of soap to create a good lather. Vigorously rub together all surfaces of your hands for 15 to 20 seconds. Friction helps remove dirt and microorganisms. Pay close attention to cuticles and under your fingernails. Keep splashes to a minimum and try not to touch the sink itself. Rinse your hands, keeping your hands lower than your elbows. Dry your wrists and hands completely with a clean paper towel. It is best to pat your hands dry rather than rubbing them. This helps prevent your hands from chapping and cracking. Turn off the faucet. Remember, don't touch the faucet with your clean hands. Use a clean paper towel. Remember these 6 steps the next time you wash your hands or assist the people you support to wash their hands. You're probably now asking yourself, how often should I wash my hands? The following are general guidelines for washing your hands. When you arrive at work, before and after assisting someone with personal care, before and after wearing gloves, before and after you eat, before and after cooking, before leaving work, after blowing your nose, touching your face or hair, after using the bathroom, after any cleanup you do, after smoking, or whenever your hands are soiled. Standard precautions requires the wearing of personal protective equipment whenever you anticipate the possibility of splashing or spraying of blood or other body fluids, such as mucus, drainage, or emesis. Personal protective equipment helps protect you from contact with blood or other potentially infectious material. Personal protective equipment includes gloves, masks, gowns, aprons, face shields, protective eyewear, mouthpieces, and resuscitation bags or other ventilation devices. Personal protective equipment must be appropriate for the task. You should wear as much as you anticipate you will need to keep blood and other infectious materials off your skin, mucous membranes, or clothing. Anytime the personal protective equipment is torn or contaminated, remove the equipment and replace as soon as possible. Gloves should always be used whenever you anticipate contact with blood, potentially infectious material, mucous membranes, or non-intact skin. When working directly with people, single-use disposable gloves should be worn. Single-use disposable gloves are just that. They should be used only once and then disposed of immediately. The wearing of gloves does not substitute for good hand washing. It's not acceptable to wear the same gloves or wash gloves between each person. Another type of glove you may use is a utility glove. Utility gloves are generally used during housekeeping. Utility gloves should be cleaned after every use with soap and water and then dipped in bleach solution. The gloves should be worn, not handled, during this cleaning and sanitizing procedure. Utility gloves are not used when working with people. They are only appropriate for cleaning. Because gloves can be torn or damaged, always bandage any cuts or open areas on your hands before applying gloves. Replace the gloves as soon as possible if they are contaminated, torn, punctured, or damaged in any way. Follow these general guidelines to determine when to wear gloves. Anytime you are handling blood or body fluids. Anytime you have open wounds on your hands. Anytime there's potential to come into contact with objects contaminated with blood or body fluids. If you are touching mucous membranes, when touching non-intact skin of any person, anytime you are cleaning contaminated or potentially contaminated surfaces, equipment, or objects. Good housekeeping helps protect everyone and is everyone's responsibility. Carefully follow your agency's policies and procedures regarding handling infectious materials. The following are some general rules when dealing with infectious materials. Always wear gloves when handling infectious materials. Handle used equipment soiled with blood or other potentially infectious materials with care. Do not let the equipment touch your skin, mucous membranes, clothing, or other items in the environment. Do not eat, drink, smoke, apply cosmetics or lip balms, or handle contact lenses where there is reasonable chance you have been exposed to blood or body fluids. Do not keep food and drink in places where blood or other potentially infectious materials are present. Clean reusable equipment appropriately before using it for another person. Single-use items should be discarded. Be careful to prevent injuries from needles and other sharp instruments when using, cleaning, or disposing of them. Never bend, recap, or break needles after use. Dispose of contaminated sharps immediately after use in appropriately labeled or color-coded, leak-proof, and puncture-resistant containers. Report any container that is full. Remember, safety razors and razor blades are also considered sharp material. Follow your agency's guidelines when disposing of sharp materials. To prevent contamination, handle soiled linen as little as possible and with minimum agitation. Gloves should be worn. Without sorting or rinsing, place soiled linens in a leak-proof bag in the area where it was used. Follow your agency's guidelines for handling contaminated linens. Transport specimens in closed containers. Wear gloves and handle the containers carefully. Specimens taken to the doctor should be placed in a closable plastic bag. Carry waste containers away from your body. Clean all blood and body fluid spills promptly according to your agency's policy. Everyone should know what to do if an accidental exposure of blood or body fluids occur, such as a needle stick injury or a splash into the eyes or mouth. This is called a significant exposure. If you are involved with a significant exposure, don't panic. Immediately wash the affected skin with soap and water. Flush affected mucous membranes, such as eyes, with water. Report the incident to your supervisor immediately. There are a number of steps that must be taken in the first 24 hours to decrease your risk of becoming ill, so quick reporting is essential. Follow your agency's guidelines when dealing with any accidental exposure to blood or body fluids. If there is a blood spill, your agency may have a blood spill kit for your use. Follow your agency's policy for using your specific kit. Infection control practices are easy to ignore because they can be so basic, but you must take the time and make the effort to consistently follow infection control procedures. Let's take a few minutes to summarize some of the main points when following standard precaution guidelines. Pathogens are disease-causing microorganisms that can be found everywhere in the environment, on humans and animals. Standard precautions is an approach to infection control that treats blood and body fluids as if they are infectious. Standard precautions includes the concepts of universal precautions and body substance isolation. It is not always possible to know whether a person is infected with a disease. The person may not show any signs or symptoms or even be aware of their infection. Standard precautions are made up of a system of safeguards designed to help protect you and the people you support. These safeguards include personal protective equipment, safe work practices, good hand washing, good housekeeping, and vaccination. All must be used together to form an effective barrier. Pathogens need to get inside of your body to cause an infection. Some ways they may enter your body are by punctures from sharp objects or openings in your skin. Pathogens can also enter through mucous membranes of your mouth, eyes, nose, or by touching contaminated environmental surfaces. In order for a disease to spread to others, certain conditions are needed. These conditions include an agent, like a virus or bacteria that can cause illness, a host, a portal of entrance, a means of transmission, and a susceptible person. Transmission of pathogens can occur by direct, airborne, vector, indirect, or droplet means. The most important and effective thing you can do to help prevent the spread of infection is to wash your hands. Standard precautions require the wearing of personal protective equipment whenever you anticipate the possibility of splashing or spraying of blood or other body fluids. Following standard precaution guidelines is crucial for protecting you and the people you support from potential infection.
So this is everyday stuff that we use to prevent the spread of infection beyond what's acceptable, okay? We talked earlier about the fact that it was the invention of the microscope that led in part to our understanding of disease, okay? Koch's postulates grew directly out of the use of the microscope to examine stuff that was too small to be seen with the naked eye. And essentially, it's a way to identify the pathogen for a particular communicable disease. The causative agent is the cause or the infection of the disease, and the postulates are a series of proofs that became the standard for determining the cause of the infectious disease, and they still play a role today, okay? Some types of disease resist Koch's postulates for a variety of reasons that we'll get into, right? Such as infectious agents that can't be isolated or grown in the lab, or those that can't be elicited in animals, such as viruses with a limited host range, and so on, okay? It's not possible to determine the cause in polymicrobial diseases, such as biofilms, right? Because it's difficult to separate out the bad actors, all right? So essentially the way that it works is postulate number one, you find evidence of a particular microbe in every case of a disease when you sample the infected material. Then you grow that pathogen, that suspected pathogen, on media, either liquid media or solid media, and then you examine it. You can stain it, you can probe it. All right, there's a variety of ways to examine on a microscope slide what you've grown. Okay, then you can take from that culture that particular pathogen, you can introduce it into a healthy organism, and you can reproduce the symptoms of the disease and then re-isolate that same pathogen from the infected material, okay? And if you can do all of those, that's likely the cause of your disease, okay? All right. So epidemiology is simply the study of the spread of disease in populations, okay? And it involves microbiology, medicine, psychology, ecology, stats, okay? And it considers all types of diseases from infectious to mental illness to addiction, okay? Florence Nightingale laid the foundation for modern epidemiology because prior to the germ theory of disease, she noticed an association between filth and disease spread. And so she was one of the first to use things like separate linens and towels for each patient, cleaning of floors, unclogging of sewage pipes. She kept notes and demonstrated that more men died of disease than died of trauma. The techniques used in epidemiology track behavior. They also use clues of the causative agent, such as the pathology, the sources, and the modes of transmission, and it tracks the number and the distribution of cases of diseases in a particular community. The results of this help public health departments generate preventative measures to reduce the spread in the population. Okay. Some examples. Okay. A notifiable disease is one that must be reported to authorities, while other diseases can be reported on a voluntary basis. It's a network of agencies at the local, district, state, and international level that keep track of these. And in your book, Table 1110 lists the reportable diseases in the US. Okay. Statistics of concern to the epidemiologist are rates of disease with regard to sex, race, or geography. The mortality rate measures the total number of deaths in a population from a disease. While the overall death rate from infectious disease has dropped, the number of persons afflicted with the infectious rate has remained high, and that's called the morbidity rate. Okay. Morbidity rate is different than the mortality rate. Okay. A point source epidemic are where infectious agents come from a single source and all the victims are exposed from that source. A common source epidemic results from exposure to a single source over a period of time, while a propagated epidemic results from an infectious agent that is communicable from person to person and is sustained over time in a population. The index case is defined as the first instance of a particular disease, even though it may not be the actual first instance, it's the first reported instance. An endemic disease is an infectious disease that has a steady frequency over a long time in a particular location on the globe. A sporadic disease is an occasional case that's reported at irregular intervals at random location. And an epidemic is an indication of the prevalence of an endemic or sporadic disease increasing beyond what we expect for the population. The time period isn't defined and it can be measured in hours or years, and the exact percent increase needed before an outbreak gets qualified as an epidemic is not defined either, right? While a pandemic, which is what we're in the midst of, spreads across continents, okay? And the reason, again, that pandemics can happen in the modern world and were less frequent in recorded history earlier is because of the invention of planes, trains, and automobiles, right? Which allows us to transport infected material or persons from one continent to the other, right? So you can see some examples here. Endemic occurrence, right? Epidemic occurrence, okay? The red dots indicating cases. Sporadic, right? Scattered, limited numbers, right? And then pandemic across continents, okay? So not a surprise, okay? All right, let's take a break and we'll come back at 3 o'clock and we will continue with our next chapter, okay? So we'll see you at 3.